Okay, good afternoon. I'm Saul Silverstein, and today I'm going to take Dr. Racaniello's place, hopefully with the same energy and fervor that he has. But I'm going to talk to you about things that I know about, because I don't know anything about what he's talked about so far. And I won't pretend, but I will answer your questions. Today's lecture is about DNA replication in viruses and the various families of viruses that Dr. Racaniello has introduced you to. If you can't hear me, move up. I can't speak any louder, and my voice is just going to get lower as the lecture goes on. So let me warn you. When you talk about DNA replication, the major problem that a virus genome faces is initiation of DNA synthesis. And that's why it's all about getting started. It's how the various virus genomes use what they have, what they bring into the cell, what they steal from the host, after all they are parasites, and how they use that material to initiate DNA synthesis. So you've been introduced to these various life forms, as I call them, and they're probably not really alive yet. But these are schematic representations of some of the virus genomes that we've been introduced to. On the top you see the Parvoviruses, and they're a family of single-stranded DNA viruses that are present as both plus and minus strands. Don't think of them in the RNA sense. These are plus and minus in a DNA sense. They don't encode. That is, they can't be translated directly into messenger RNA. They must be transcribed first. And for that to occur, they must become double-stranded DNA molecules. These DNA virus genomes are represented by this very unusual panhandle structure at both ends of the molecule. And that occurs because they contain terminal repeats and inverted terminal repeats. And we'll get into that in a lot more detail later on. And I just want you to get a flavor for what we're talking about. We'll also be talking about one very large viral genome, the herpes virus genome. And in particular, we will be discussing herpes simplex virus because it's perhaps the best studied of the family members. And it has a very unusual genomic structure, and we'll address that a little bit later. But the thing that is of interest most in terms of what we're talking about is the presence of 3 different origins for DNA replication. And they are used under different circumstances. We'll also talk about the linear double-stranded adenovirus genome, and it's of interest because it has a 5' terminal protein that's stuck at both ends of the virus genome. And in endless detail, we will discuss replication of this circular molecule. So, why do viruses undergo DNA replication? I think the answer, or the immediate answer, is pretty obvious. They have to replicate these genomes to make new progeny. Everything is about getting the progeny out. It's certainly true in everything about life. And this almost always requires the expression of at least one virus protein. That means that the virus genome must be transcribed at some point. In some instances, many virus proteins are elaborated. DNA, however, is always synthesized in the 5' to 3' direction, and it's done in a semi-conservative replication fashion. Both strands are replicated. Replication initiates at a defined origin using a primer. And the amazing thing is that these primers can be very variable. They can be DNA structures. They can be Okazaki fragments, those small RNA pieces that are made by primases and polymerase alpha. Or they can be proteins. And we will discuss examples of each of those. But most of all, remembering that this is a parasite, it's the host that provides other proteins that facilitate and allow for this very complex and exquisite replication machinery to work and to function in cells. Now, as I've shown you, and as you've heard from Dr. Racaniello, genomes come in a wide assortment of shapes and sizes. They're just like people. There are many different flavors. Replication yields progeny. And replication is also a very important point in the life cycle of a virus because it acts as a switch for gene regulation. There are almost invariably a family of genes that are transcribed and translated prior to DNA replication. And those, depending upon how they're utilized, are called either immediate early or early genes. And they, among other things, encode for proteins or functions that allow for the virus to replicate its DNA. So that's a very important regulatory event, and it separates the machinery that's used to activate genes and to activate replication, generally speaking, from the machinery that's used to make capsid proteins, proteins that are required in large abundance to encapsulate and enclose the various virus genomes. It's always delayed after infection. It's never the first event, with one exception, because there's always an exception. So what are the outcomes of DNA replication? Usually we think of lytic infection as being one type of infection, and there it's quite simple. Quiet. So we get new progeny and lots of little virus genomes, and we get high copy number. Sometimes thousands and hundreds of thousands of genomes are made in an infected cell. The other type of infection, which is in some ways much more interesting, is the latent infection. And that's when a virus enters a cell, and we'll talk about this in a lot more detail later in the course, and sort of hangs out. It finds a stable environment, a niche where it can be very, very happy, where it can maintain its chromosome at a low copy number and stably in assimilation with the host. And in some cases, such as the Epstein-Barr virus, it replicates with the host. And it has an entirely different replication cycle in latency versus lytic infection. Come in. These virus genomes can be episomal, that is, they're extrachromosomal, like plasmids are in bacteria, or they can be integrated into the host. These pose different problems for when a virus is reactivated, and that occurs in almost every instance of latency. So viruses go from latently infecting cells to entering the lytic infectious stage and producing new progeny. The triggers for those are some of the most interesting questions that we ask in virology. Now, what are the requirements for DNA replication? You require a specific origin that is recognized by either host or virus plus host proteins for initiation of DNA replication. When the sequence is DNA, it's almost invariably an AT-rich sequence. And it's an AT-rich sequence because AT base pairs are preferentially or more easily opened, melted apart by helicases. And that allows access of polymerases and initiation factors and proteins, be it as they may, to this segment of DNA to begin copying. Priming can result from RNA, in which case you have those small RNA-DNA hybrids that are termed Okazaki fragments of about 140 nucleotides. From DNA hairpin structures, where the DNA bends back on itself, exposes a free 3' hydroxyl, and allows for progressive synthesis of DNA molecules. Or it can be protein that's covalently attached to the 5' end and allows for the initiation of DNA synthesis. The next event is pretty easy. It's elongation. And while there are different virus proteins that help elongation, and we'll talk about those in a bit, by and large, it's the polymerase chugging through the molecule, making its way through hairpins, making its way through DNA structures, melting things as it goes, and there are a lot of ways in which it can do that, at a fairly constant rate. And finally, there's the end, or termination. And termination can be a very specific event, or it can be a random event. That is random in terms of the fact that there is no specific nucleotide sequence that is recognized by a polymerase or a polymerase complex, but yet two replication forks meeting at one point. And we'll see that when we talk about replication of circular molecules. Okay. other requirements. Viruses don't replicate well in quiescent cells. Neurons, terminally differentiated cells really aren't dividing. And because they're not dividing, they're missing a good portion of the equipment, of the machinery that a virus needs to replicate. So, for example, a neuron has no ability to replicate DNA, it can repair DNA. And as a result of that, it's going to be deficient in certain DNA polymerases, certain kinases, ligases, all of the enzymes that are really important for DNA replication. So what does a virus do? It's clever. It induces host replication enzymes, and it induces cell cycle regulators. These proteins are important for initiating DNA synthesis of the host. Now, that doesn't necessarily mean that the host goes on and replicates its own DNA. In fact, in many cases, infection with a virus abrogates host DNA replication. But because the virus comes in and brings these handy little proteins of its own, or because they are translated and, at very early times post-infection, they induce host replication enzymes. And this results in activation of the cellular DNA synthesis machinery. Machinery is localized, with the exception of the pox viruses, to the nucleus. So you take a cell that's quiescent, is not actively synthesizing DNA, and if you activate the cell machinery, you now provide landmarks and points within the cell for which a virus can dock its DNA, dock its genome, and initiate the replication process. And indeed, these are a very limited number of sites in which a virus genome can do this. And then, again, we get to the point that the virus must elaborate immediate early and early gene products before it can begin initiation of DNA synthesis. So what's the host for? The host is a bag. It's a bag of enzymes. It's a bag of structural elements. And these enzymes and structural sites, mostly present in the nucleus, provide a place for the virus to lodge. Simple viruses conserve genetic information. So they usually elaborate a protein or two at most, and they use the host. In that case, the virus can replicate along with the host. The host will have some limited DNA replication, and the virus will also have DNA replication. With the more complex viruses, however, they encode many, but not all the proteins that are required for replication. So we can put virus proteins into a test tube along with either a plasmid that contains a viral origin of DNA replication or a viral genome and get some replication. It's very difficult to get initiation. It's not precise. And the reason for that is that we don't know all of the components and we don't have the structural motifs that are required for assembling the genome in the proper place within the cell. But what we can do is we can add back virus gene products to cells and allow the cell to have only a limited number of virus gene products and ask if those proteins plus an authentic origin will allow for replication. And we'll end the lecture on that note. So, where does the replication polymerase come from? As I said to you before, small DNA viruses do not encode an entire genome replication system, but they do encode proteins that orchestrate the host. So members of the Papillomaviridae, the Polyomaviridae, and the Parvovirus, all small DNA viruses, double-strand circles, single strand, they use a lot of the host machinery, and they tend to encode 1 to 2 proteins. 2 proteins for papilloma, 1 protein for the Polyomaviridae, 2 proteins from the Parvoviruses, although they're really just one is a subset of the other. Large DNA viruses encode most of their own replication systems. So the herpes viruses, the adenoviruses, and the poxviridae all have polymerases, helicases, topoisomerases, primases, you name it, they got it. And here they are. DNA polymerase and accessory proteins, among them a primase. The primase is important for initiating DNA replication. Things that I may refer to as OBPs, for origin binding proteins. Specific proteins that recognize a site for initiation. Helicases, to open up the DNA, to melt it, to allow that first set of nucleotides in. Exonucleases, to pair things back and repair. And other proteins such as thymidine kinases, ribonucleotide reductases, or DUTPases, to make precursors for nucleotides. So a quiescent cell doesn't elaborate these enzymes. So you can't have TTP. You can't make DNA if you don't have thymidine triphosphate. So these guys will bring things in, or they'll convert nucleotides that are present in cells from ribonucleotides to deoxyribonucleotides. And this helps to get the machinery in order for the virus to replicate DNA. Now, that was not so good. Okay. Replication occurs where? In replication centers, of course. And these replication centers are present in the nucleus. So what you're looking at in this panel and this panel is the nucleus of a cell, stained for its DNA. So DNA templates and replication proteins associate at discrete sites within the nucleus that are called ND10s for nuclear domain 10s. Why? Because there are about, on average, 10 of these inside the nucleus of a cell. And what happens is virus genomes assemble at these ND10s. And it's here that they initiate DNA replication and transcription as well. The sites are preferentially used. So there might be 10 of them, but they're not all equal. And we don't know what controls access. They can all be occupied, but replication occurs at different, with different affinity at different sites. Now, in this panel is a antibody that's staining for a particular virus protein. It happens to be a protein from herpes simplex virus, which is a single-stranded DNA binding protein. In the cell without virus in it, of course there is none. And in the cell that has virus, you see that the protein assembles where DNA assembles. And if you were to use this probe as a probe for viral DNA, you would note that that's where the viral DNA is. And when you merge these two images, you see that they overlap. So the DNA templates and proteins that are important for replication of the DNA genome are present in these nuclear domain 10s. Okay. In order to get started, the virus needs to find a place to start. So what's the starting line for a virus? Frequently, it's these AT-rich DNA sequences, and they are recognized by as a viral origin by specific proteins such as origin-binding proteins. And these proteins are sequence-specific. It's not a structure that they're seeing, but rather a sequence. There are instances of structure-dependent initiation, but they work through a different mechanism. And what they do is they seed the assembly of a multi-protein complex. So somebody comes in, recognizes the site, and that protein has a surface that's exposed. It's engaged with DNA, and once it engages DNA, it changes its shape as a result of that engagement. And that exposed site allows for interaction with other proteins. And what you do is you build up a multi-protein complex, and you can then initiate from that. Some viruses have one origin, others have up to 3. And as I said before, they're used for different purposes. One thing that has become very clear is that these origins of replication are often associated with highly transcribed control regions. And that feature is important because what it tells you is that these areas are very active areas, so they're naked areas. They're available for proteins to assemble on them. So they're not highly chromatinized in the instances where the virus forms a chromatin-like template. And there are viruses that don't have nucleosomes or features of nucleosomes, but have a chromatinized like genome that is areas are covered by histones and various accessory proteins. So we're back to our genomes. And once again, we're going to start here with the Parvoviridae. We're not going to actually start with that. And the initiation site for the Parvoviridae is right here. Or here, depending upon whether this is a plus or a minus guy. For the simplex, it can be in these origin S sequences, which are a complicated portion of the virus chromosome, or down here in a latently infected cell. In the adenovirus, it starts with a protein which sits opposite the 3' end and binds and allows for polymerization through. And in the Polyomaviridae, there is a unique origin in the circular molecule. And finding that is difficult because the molecule doesn't really look like this, but rather it looks like this. This molecule that is just twisted up on itself. And if you introduce a nick anywheres in this highly super twisted molecule, then you'll open it up and you'll get what's called a relaxed circle. So the highly supercoiled molecule is form 1 and the relaxed form is form 2. How many of you can see the rubber band? Would it be better with the lights on? Maybe. Okay. If you take a rubber band and you pinch off the top, then it remains just the way you see it, as a rubber band. As replication proceeds, though, the top gets pushed out, DNA gets extruded through a machine. We'll talk about that in some detail. And behind it, because this part is unwound, behind it, it tends to curl up like that, and it gets all knotted on itself. And that's a supercoil. And the only way to release that is with an enzyme called the topoisomerase, which puts a nick in DNA and unravels it like that. So those are some of the real problems that are associated with DNA replication, and we'll try and address them as well. So here's your supercoiled DNA molecule. It starts as a circle, and what happens is it gets all twisted up on itself, and the more twists that are introduced, the more compact the molecule becomes, until it's a very, you can think of it as a ball of string almost. It's a very tight coiled subject. A single nick in that molecule, however, by topoisomerase, will push it all the way back to this relaxed form. Open, closed, compact, spread out. So what does an origin look like? Well, not exactly like this, but somewhat like that. And what you have here is a schematic representation of a family of, I don't have this down pat, of a family of origins from different virus genomes. SV40 is a member of the Polyomaviridae family. And what it has is this AT-rich sequence here and another one here. And what you see is something called LT binding site 1 and binding site 2. LT stands for large T antigen. It's the immediate early gene product of this virus. It's the only protein, for all intensive purposes, that's made right after infection and entry into the nucleus. So it's transcribed before DNA is replicated. And that protein is required to initiate DNA synthesis. And what happens is T comes back and it binds right at this site, which is adjacent to the early promoter, and it helps to open up that sequence and make it available for DNA replication. Herpes simplex virus has an origin that's a little bit more complicated. It's a very, very large palindromic sequence, and it is very AT-rich. And it's AT-rich in the context of the virus. The virus is about 68% G plus C, and this region here is 40%. So that's very AT-rich. And that's recognized by its origin binding protein. And much like SV40, the origin is placed between two transcriptional units, again allowing access. And for adenovirus, we have an entirely different picture. We have its double-stranded genome. The 5' end of it has something called terminal protein. 3' end is accessible, and what happens is it recognizes TP and puts on a molecule, which we'll talk about later, and that's how it initiates its replication. So, what are the various recognition proteins? How do they function? Polyoma T antigen binds specifically to DNA. It is a site-specific DNA binding protein. Papillomaviruses use two proteins, E1, which has helicase activity and is absolutely important for origin recognition, but doesn't bind specifically. What it requires is another protein elaborated by the virus called E2. E2 binds to a specific sequence in Parvovirus DNA and allows E1 to come in, and then you can start initiation. So it's a two-pronged attack.
So E1 comes in first?
No, E2 comes in first because E1 is just spaced out, all right? It doesn't know where it's going. But once it has E2 available, it can find where it needs to be. The parvoviruses have a protein Rep68 and 78, and I said to you before that there are two proteins, and what they are is one is a subset of another. But what they do is also bind at the ends of the single-stranded DNA and the double-stranded molecule, and they unwind the DNA, allowing for access. It's interesting because this protein is also involved in terminal resolution, and you don't know what that is, but we'll explain that as we go forward. The terminal protein in adenovirus begins as a pre-terminal protein, PTP, and that binds at a terminus and it recruits DNA polymerase. And then DNA polymerase has a very specific function for adenovirus, and that is to place a deoxycytidine moiety onto a serine of PTP, and that cytidine is actually the primer for DNA replication, initiation of DNA synthesis. Herpes viruses use protein UL9, and that's an origin-binding protein, again site-specific, and it recruits a bunch of viral proteins to AT-rich origins and then unwinds DNA. So, just to give you a little more feel for where we're going, again, we're going to come back to our genomes. And you remember that we have double-stranded DNA. It can be transcribed into mRNA, and it can be replicated into double-stranded DNA and go into the particle. The polyomavirus genomes are circular and supercoiled, so they're all twisted up on themselves. We have here an adenovirus genome, the very large herpes virus genome, and the Poxviridae, which we're not going to talk about because that would require an entire lecture to even begin to explain them, but they're actually just a big circle. All right? They're double-stranded, and the ends are covalently closed. Okay? So, that's the double-stranded guys. What about the single-stranded guys? The single-stranded guys are, generally speaking, pretty easy to understand. You either get a plus strand, and it gets made into a double strand, which subsequently can be single or double-stranded and packaged. Or you have this guy, the Circoviridae, which Dr. Racaniello mentioned, which is a single-stranded genome in only one configuration. So, if you think about it, it has to use the host. The question is, how is DNA synthesis primed? What's the origin for DNA replication? And that's an unknown. And this is of some interest because it turns up in all sorts of different products. So, for example, there were a large batch of purified protein that's used to treat, I don't remember which disease, and was found to be contaminated with circovirus DNA, or virus particles. And so all that stuff was thrown out, millions of dollars worth of product. And the question is, why? We have no idea what this virus does. It doesn't seem to have caused any pathogenesis. Where did it come from? Probably from some serum component that was used to grow the cells. But without knowing what it is, there's a reasonable doubt, and so you get rid of it. And we have our Parvovirus, which start as single-stranded molecules, get replicated to make a double-stranded DNA molecule, and keep doing this back and forth, and we'll show you what that's all about in a bit. The last one, and We're not going to talk much about it, but this is hepatitis B, which is thankfully pretty much under control because of good vaccination protocols. But this is a very interesting molecule because you'll note that it's sort of double-stranded, and one strand is complete and one strand is incomplete. And the incomplete strand actually overlaps the other strand. So what you have is a 5' end that has to be cut because this guy is going to replicate further. So the first thing this virus does, or this virus DNA does when it gets into a cell, is to use the host to repair this molecule. And then it becomes a supercoiled, circular, covalently closed DNA molecule. Then it can be transcribed and begin its process. But the most interesting fact is that after it does all that, it's really a retrovirus. So you'll learn more about that as we go through the retrovirus life cycle. So there are two basic modes of replication. There is replication fork-based synthesis, where there is a leading strand, where DNA replication elongation is continuous, starts at with a specific primer, and just reads through. And there's a lagging strand. And the lagging strand is initiated with RNA molecules, and it is done discontinuously. And viruses such as papillomavirus, polyomavirus, herpes viruses, and even the retroviruses, at least in the proviral form, use that mechanism. Adenoviruses and parvoviruses and poxviruses with their hairpin use a strand displacement mechanism. That is, they displace one strand as they replicate another. Here's your parental strand. Here's your other parental strand. This one gets displaced as new DNA is laid down. Then the other one gets replicated. And we'll go through that for adenovirus. Okay. Let's start on our replication journey with polyomavirus replication forks. As I said to you before, initiation is from a single origin, single site, sequence-specific recognition by the T protein. T actually stands for tumor antigen, and it's because part of it gets elaborated on the surface of cells that are transformed by this virus. And that's an early protein. It's the first protein made in the infected cell. These molecules replicate as covalently closed circles. That is, they remain circular during the course of replication. And that goes back to the rubber band. Part of that circular molecule is actually extruded and is relaxed and is held, and the extruded part is held in place, sort of like when you ride a horse and you gallop and you hold your two hands together with the reins. We'll show you about that in a bit. Leading strand replication occurs via extension from an RNA primer, and the lagging strand replication can't start until the replication fork has moved some, until that leading strand fork has moved. It also uses RNA primers, but it creates discontinuities because it's replicating in the same direction, and the only way it can do that is by essentially replicating backwards. And so you're left with a conundrum of how do you fill in the gaps? So to talk about polyomavirus replication, you need to know something about the properties of T antigen. And you'll also need to know something about these properties when we talk about transcription, because this is a very diverse molecule that provides for a great number of functions during polyomavirus replication. As I said, it's a DNA binding protein. Perhaps the most fascinating feature is that it's species specific. So a polyomavirus that originates from a mouse cannot replicate in human cells, and vice versa. And the reason for that is that pre-initiation complexes do not form in the wrong species. So if you have a human virus, you put it into a mouse cell, it gets into the cell, it finds the nucleus, and it does squat. It just can't do anything because it can't initiate DNA replication. And the reason for that is it doesn't interact with the host DNA polymerase alpha primase protein complex. So it can't initiate. This protein binds and sequesters cell cycle regulators. Why? Because it causes cells to enter the S phase. So why is that important for these viruses? Because once they enter S, they synthesize DNA, or they gear up the machinery that's used to synthesize DNA. So it becomes available. Its synthesis is autoregulated. That means it turns off its own synthesis. It makes a large amount, then it shuts itself down. The protein is heavily modified. That means it's decorated with other moieties such as phosphates and ribosyls and adenosines and things like that. And it's the decoration that controls its ability to bind DNA. And the various levels of decoration also control the cooperativity of the molecule. That is, the molecule actually forms hexamers. So there are 6 T molecules in each active replication fork complex. And it is this cooperativity and protein modification that affect unwinding of DNA, opening up the helix. And the best work on this has been done actually by Carol Privis here in the biology department. And so here's T antigen as a schematic. It's a large protein, 708 amino acids, and it has all of these various binding, all of these various protein domains that are important for interaction, for example, with polymerase alpha of the host, p53, a cell cycle regulator. It has an ATPase activity. It has single-stranded DNA binding activity. It's a helicase. It opens up DNA. It binds to the origin of DNA, and it interacts with proteins such as the retinoblastoma protein, another important cell cycle regulator, and cell heat shock proteins, which are important for how proteins fold and what they interact with. So this is an enormously complicated molecule. It has many different sites that are phosphorylated, and it's these sites of phosphorylation and other modifications that control how and what the molecule is doing at different stages in virus replication. Okay, we're back to the core sequence, and this is where the virus initiates its DNA replication. This origin is between the promoter for the early region and the promoter for the late region. And that's because this is an area that opens up and is nucleosome free. And it's nucleosome free so that there's accessibility. This supercoiled DNA molecule is covered with histones, and it looks for all the world just like a normal chromosome. So that it's totally wrapped up around nucleosomes. There are a bunch of binding sites that are adjacent to this core or within it, and they're the sites for binding of T antigen and also for transcription activation, and it's AT-rich. So we've laid out the circular molecule as a linear form with its single origin, and what I can tell you is that replication is bidirectional. So it's proceeding in both directions at once. Starts at a single site, goes around the chromosome in 2 directions. As a result of that, you have 2 replication forks. One going this way and one going that way. And the origin has now been duplicated, and you have what are called leading and lagging strands with the 2 replication forks. What's the difference? DNA synthesis on a leading strand is continuous. It doesn't have to stop for anybody. DNA synthesis on the lagging strand is discontinuous. It's a series of short polynucleotide sequences that are primed by RNA, extended in the same manner that leading strand synthesis is. But because it's going backwards, it runs out of room, and we'll see how that works. And then you have a problem of putting those pieces together. So, what happens when you initiate? Here's our origin of DNA synthesis, and here is our hexameric large T subunits, and they bind ATP. Once they assemble on the chromosome, as I said before, you think of it as 2 fists, and they're right next to each other. What they do is they distort that palindromic sequence which is present in the core and begin to unwind the DNA. And that's what's represented here. And what happens is that DNA gets extruded. It's now a single strand, one in one direction, one in the other, because remember, we have 2 replication forks. And, excuse me, a host protein called RPA, or replication protein A, now assembles on the single-stranded DNA. So it's a single-stranded binding protein. And this, in the presence of a topoisomerase, remember that topo? Everything's wound up, so that'll nick and allow things to unwind. Now allows for the extrusion of the DNA and makes it available as a template for DNA replication. So you now have the origin exposed and it's available. Now, how do we know there's a single site for DNA initiation? This is a great experiment. This is one of those, picture is 1,000-word guys. And okay. And it's nice to think about. If you assume that a circular molecule has a single site of initiation, and that there's also an asymmetrically positioned restriction endonuclease site, then when you open up the molecule, the origin of DNA replication will be asymmetrically positioned from the ends. And that's shown down here. Look at that. That little bubble is the origin. And you can see that its distance from the origin to the left end versus the right end is quite distinct. Again, here, it's better shown here. That's the origin, that little bubble. And this is one arm. That would be this long one. And this is the other arm, and that would be the short one. Okay? Now, as DNA synthesis occurs, and as DNA elongates, this bubble gets bigger. Gets bigger because DNA is replicating. And eventually it begins to expand until it occupies the entire sequence up to the restriction site. Once you pass the restriction site, it gets cut, and you have two molecules that are dangling off. So now, how do you know that replication is bidirectional? Anyone? Don't all shout out. What tells you that it's bidirectional? Yes?
The size of the bubble is getting bigger, so it's like opening up.
Right. The ends are getting shorter. It's growing in both directions at once. Yes?
What do you mean by the ends are getting shorter?
Well, watch. The distance from the bubble to this end gets shorter as replication proceeds until you get to the point where it's really close. Now, once you get to that edge, so what you have to remember is that this is a molecule caught in the act of replicating. It's sort of like a bandit, right? And then you cleave it. Now, if that site, the restriction enzyme site, was replicated, so where the green dot is now here, those ends would now be open because they'd have been duplicated. Okay, so back to replication. All right, we now know that it's bidirectional. We know that it starts at a unique origin. And now we have the problem of leading and lagging strands. That is, remember, DNA synthesis only occurs in one direction, 5' to 3'. So if you start with your leading strand and you replicate here continuously, how do you replicate this part of the replication fork? And that's the issue. Well, you do it using Okazaki fragments. Okazaki fragments, as I said before, are RNA-primed DNA polynucleotides. So what happens is, once this leading strand begins to be synthesized, then the polymerases, and we'll go into this in nagging detail in a second, now have available to them parts of this replication fork. Using polymerase alpha as a primase, and T antigen is sitting right here and here, by the way, you now lay down a small track of ribonucleotides, which have 3' ends that serve as primers for initiation of DNA synthesis. So DNA synthesis starts here and goes to there. And then it starts again further upstream. It can only go there because you've already synthesized that piece. The problem then becomes putting these guys together. And they're put together because, remember, there's an RNA and DNA part, and what happens is the RNAs are removed by a specific exonuclease. DNA is repaired, ligated, and filled back in. So the leading strand is easy. a pre-synthesis complex between polymerase alpha, one of the host DNA polymerases, T antigen, and replication factor A forms on the template. Replication factor C binds the product of polymerase alpha at the 3' hydroxyl, along with a protein called proliferating cell nuclear antigen. And this is a clamp. That is, it's a ring structure that sits on the replicating DNA molecule. And in order for that clamp to get on to the DNA, it requires replication factor C. So C is called a clamp loading protein. It actually allows access of pCNA and polymerase delta. And polymerase delta is the guy that's responsible for extending the DNA chains. So once pCNA is on DNA, it releases polymerase alpha, and polymerase delta can do its business. RFC and pCNA form sliding clamps along the DNA, and then you get continuous copying of the parental strand. Don't worry, I'm going to show you what that is. The lagging strand is not so easy. The first primers and Okazaki fragments are made by polymerase alpha primase complex. Then the DNA is copied from the replication fork back towards the origin. That's why I said it's going backwards. It's going in the right direction, but it's starting further on up the turnpike. Multiple initiations are required to replicate the template strand. Unlike the leading strand, the lagging strand requires many, many starts. Both leading and lagging strands move in the same direction. And then the question becomes, which moves, the DNA or the complex? And it turns out that the template has to move, otherwise you can't unwind this mess. So the actual complex, where T antigen is and where the replication forks are held in a stable configuration. So how do we know things unwind at the origin? Well, when you look at them, when you catch these molecules in the act, these are T antigen hexamers, 1, 2, and the DNA gets looped out. It's single strand, and we know it's single strand because this has been visualized with the aid of a protein that recognizes single-stranded DNA, a single-stranded binding protein. So it only binds to the single-stranded area of the molecule. So the part that's not in the process or being prepared for replication binds very little of the protein because it's double-stranded. Okay. What are the host factors, proteins that are required? Can you see this? I'm just going to elaborate them. We'll go through them quickly. Replication factor A, it binds to single-stranded DNA, origin unwinding in cooperation with large T. DNA polymerase alpha, primase, that's required for synthesis of RNA primers, the beginning of the Okazaki fragments, and it's used on both the leading and the lagging strand. RFC is a ATP-dependent clamp loading protein. And it's also required for not only engaging pCNA, but releasing it from DNA. pCNA is the sliding clamp. It's the structure that allows the polymerase to get to it. Polymerase delta is the enzyme that provides for processive synthesis of both strands when bound to pCNA. So this is the DNA polymerase. And then we have another couple of characters in here. There's RNase H, which removes the RNA and the RNA-DNA hybrid, so that part of the Okazaki fragment, which is RNA. And then there's a protein FEN1, which is a 5' to 3' exonuclease, which also is involved in removing RNA primers. And finally, DNA ligase. There are actually more proteins, but enough is enough. Okay, so this simple diagram shows you how DNA replication is bidirectional. And it's really not that hard. So I've broken it up into two parts. And the first is the leading strand. And let's just follow it, because all I'm doing is recapitulating what we've spoken about for the last 10 minutes. It gets easy. You look at the pictures, they're pretty, it's cool. All right, so we have two large T hexamers, and they've extruded DNA. DNA is single-stranded and bound by RPA. Polymerase alpha primase, this little guy here, the olive green and purple guy, comes in on the leading strand, recognizes the origin, and along with deoxynucleotide triphosphates and NTPs, because you need NTPs to make ribonucleotides, it synthesizes this RNA-DNA hybrid. Once that first Okazaki fragment is made, RFC binds the 3' end of the RNA-DNA hybrid, okay, because you've got one strand that has a little bit of RNA in it and a little bit of DNA. So the 3' end of that is bound, and pCNA is added on in the presence of ATP. So now we have this structure, which is the loaded clamp. Once that occurs, polymerase alpha, which started the whole thing, is knocked off, and polymerase delta comes in, and in the presence of deoxyribonucleotide triphosphates, it synthesizes new DNA. So now we're making double-stranded DNA in this direction and in this direction. That's the leading strand, and the leading strand is elongated. Now, once this opens up, then it frees the lagging strand, or it makes the lagging strand accessible to the same family of proteins, polymerase alpha, and they can put in their RNA initiation nucleotides. So you get your RNA-DNA hybrid molecule, and it starts there, and it's running in the same direction. And of course, it runs out of room because it's already double-stranded on the leading strand. So you start here, you go to the origin, you stop. And you start here, you go back to where this was made, and you stop. And you start here, and you continue in that process. So it's really an ungainly way to do something because it's a constant series of stops and starts, but it's the way you make the other part of the molecule. In this, yes? No, this is all going on at the same time. The only thing is a little bit of a lag in getting the lagging strand started, which is originally why it was called lagging. At first we didn't know that it was discontinuous. We weren't smart enough to figure that out. And then a series of host enzymes come in, RNase H, FEN1, and ligase to complete this double-stranded molecule. So you end up with removing the RNA, filling the gaps, and sealing it. And if you look at it at the replication fork, it looks like this. Here's your T antigen hexamer. Here's the parental DNA, right? This is what you start with. There's a little motor in there, a helicase, which opens everything up, separates the strands, and makes them available for DNA synthesis. One is continuous, that's the leading strand, and one is discontinuous, where small loops of DNA are pushed out, and they get replicated, and then the next loop comes along and gets pushed out, and they get finished in that way. And to show you what, let's see, can I start this from here? Okay. Can you hear that? If not, let's kill it. What's important here is to realize that here's the parental strand coming in. It gets unwound by this furry blue molecule. And it pushes off a leading strand where DNA is made continuously, and a lagging strand where it loops out the DNA as a template, allows for formation of that Okazaki fragment, fills that thing in, and then adds it back to the molecule that's being replicated. Oops, didn't mean to do that. Okay, now you're left with the problem. You've made DNA, you have a circular molecule, and if you make DNA from a circular molecule, what do you get? Anyone? You get another circular molecule, right? So you have two circles. And the problem with these two circles is that they're actually intertwined. So you have to separate daughter molecules from the replication complex. And that's where enzymes such as topoisomerases come in. They relax these supercoiled molecules, and they unwind them. And what that does is relieves torsional stress in the molecules and permits them to come apart. Yes? It only cuts one strand at a time. So it holds it intact. It has a way of cutting and then cutting. And even when it cuts two strands, so there are topo 1s and topo 2s and these different guys, they hold the molecule together. Because if they didn't hold the molecule together, you'd never get that circle back. Okay? So, and in fact, that's a more complicated issue, which I didn't want to get into, but that's your answer. you're left with two molecules that are intertwined, and those are called catenates, molecules that are wound up inside of each other. And you have to separate them out, and that's what the topo 2 does. It cleaves and reseals through hydrolysis of ATP. So what does that look like? As you look at replication, here's your replication fork. And remember the top of the rubber band. Top of the rubber band was just hanging there like a rubber band, just like the rubber band you always knew and loved. The bottom part got all tangled up because when you released the energy here and you allowed it to unwind, it transferred the energy down through the rest of the molecule and caused it to twist up on itself. So that part was overwound, and those are called supercoils. And eventually you end up with something that looks like this, except this is all twisted up, and as this grows, it gets bigger and bigger. Strand 1 and strand 2, think of them as thing 1 and thing 2 from Dr. Seuss. And you get this molecule, which is a catenate. It's two circular molecules that are wound up inside of each other, and they're actually supercoiled. And what the topoisomerase does in all of its glory is separate the two molecules. So you get a blue molecule and a red molecule. Of course, they're the same. How else can you prime DNA synthesis? You can prime it using a specialized structure. And the specialized structure in the case of parvoviruses is itself. So it self-primes. It forms a template primer. The genomes of parvoviruses are both plus and minus strand, and they contain inverted terminal repeats. And as a result of that, here's the end of the molecule, the 3' end of the molecule. There is a sequence within 100 base pairs of the end that's identical, but the exact inverse. So it's complementary. That allows the formation of this hairpin. And this hairpin has this very lovely 3' hydroxyl, which is a great place to start if you have something to copy. And what have you got to copy? You can copy yourself. I could make some dirty jokes about it, but I won't. So this virus is called a dependovirus because it depends on the host in order to replicate. It doesn't use polymerase alpha because it doesn't need the RNA primer. Instead, it uses its inverted terminal repeats as the primer for DNA synthesis. But it uses all the other friends that we talked about, polymerase delta, RFC, and pCNA. It also needs 78 and 68 proteins that are elaborated shortly after infection, and they form, they provide two functions. One is the initiation of DNA synthesis. The other is resolution. That is getting those single strands back out, providing new templates. So they have endonuclease activity, a helicase activity to unwind, and they bind the 5' terminus of the genome. There's no replication fork in this guy, but we're going to have semi-conservative replication. Why? Because we have plus and minus strands, and we'll show you how that happens. So here's a parvovirus DNA molecule, single strand, and it has not only those inverted terminal repeats, all right, but it also has these larger inverted terminal repeats. So the 3' end and the 5' end are the same, except they're backwards. In fact, they're complements of one another. They could form a structure like this, or the one that we've been showing you all along, where the ends hairpin out and form this structure. When you have this structure, you have a natural template for DNA synthesis. So the molecule could be elongated. Blue is parental, red is de novo synthesized. When this molecule forms, Rep 7868 recognizes this sequence, and it cleaves it. Now look what you've got. You've got the Rep protein sitting at the end of the molecule, okay, at the 5' end of the molecule, and the molecule is part new and part old, something borrowed, something blue, okay? So what we have here is a hybrid molecule, parental and daughter, and the end of the parental strand. Once this is nicked, it's released, extended out. This end can be filled, and now you have two strands, each of which is a hybrid molecule, that is part new, part old. But if you look very carefully, what you notice is that the sequence at the ends, which went from A, B, C, A, D, is now ACBAD, and it's a function of this having folded over on itself. And what happens is each time you replicate it, you get an iteration of it, and you get a change. When this protein binds, when 7868 binds, it allows for displacement of this strand. And this guy can now fold back over on itself, as it did before, and you can continue synthesis again. So each one of the hybrid molecules, part parental, part daughter strand, are capable of being replicated, and they replicate exactly in the same way, and you get thousands and thousands of genomes in a cell, single-stranded, and both plus and minus strands are packaged. So that's pretty cool. Another way of initiating DNA synthesis is through protein priming. And the adenovirus uses that. And it uses that precursor to terminal protein that I alluded to before, PTP, and adeno-DNA polymerase. So adeno encodes its own DNA polymerase. And before it can replicate DNA, it has to make PTP and polymerase. It also makes a bunch of other proteins, but we won't get into those today. Adeno polymerase links the alpha phosphoryl of DCMP to a hydroxide of a serine in PTP. It's only added, that is, this DCMP is only added when the protein primer is assembled with polymerase into the pre-initiation complex at the origin. So it doesn't just get randomly added to the protein, it only happens when that protein is recognized DNA. That allows for the 3' hydroxyl to act as a primer for the synthesis of the daughter strand. So that tells you immediately that the other strand has to end in a G, because it's got to be complementary. And then you get semi-conservative DNA replication from different replication forks. We'll show you how that works. So here's our adenovirus genome. 3' end of the template strand. Here's your 5' to 3', 3' to 5'. 5' end is bound by terminal protein. Why? You'll see in a second. What happens is the 3' strand recognizes this complex, invades along with adenopolymerase, and then once it's seated, the polymerase places the deoxycytidine moiety onto the serine of PTP. Another virus early protein that's made is called the DNA binding protein, and that's a single-strand DNA binding protein. It's made in enormous amounts. And what happens is that as this molecule extends DNA, the other strand gets pushed out, and that single strand gets coated with DNA binding protein. So that's the displaced single-strand template. This guy finishes its replication. So now you have semi-conservative replication, right? Daughter, parent. And the strand that is displaced is now covered with DNA binding protein. The ends anneal. The 3' end is recognized by PTP and DNA polymerase, and it starts the whole cycle over again, and it replicates the other strand. So what you have is discontinuous semi-conservative replication. So it's strand displacement synthesis. Adenovirus uses a protein primer as opposed to a DNA primer or an RNA primer. There are origins at both ends. Why? Because this molecule sits down when it recognizes a 3' end, and any linear molecule has two 3' ends. Any duplex linear molecule has two 3' ends. You get the assembly of the pre-terminal protein into a pre-initiation complex. That activates the polymerase to covalently link the CMP to the serine residue in PTP, and then you get semi-conservative DNA replication from different replication forks. Now, life is not always that simple. Sometimes it's even more complicated. And viruses of the herpes virus family, or most of them, have multiple origins of DNA replication. And herpes simplex virus has two identical origins that are located in a portion of the virus genome known as known as the S sequence for short. So there's a short and a long sequence. And what I will tell you, and you don't have to, you're not responsible for this, but you should think about it because it's a mind bender, is the fact that the junction between these sequences, short and long, acts as a molecular swivel. And so you can get molecules that have long running this way and short running this way, long running this way and short running this way, inversion of short, inversion of long, and inversion of both. So there are 4 different forms that are present in any population of virus molecules, and they were equimolar, and we don't know why. We're not even really sure how they occur. There are lots of theories about them. Most of them are just that, they're conjecture. But the DNA does enter as a linear molecule from the virion, and shortly after it enters the nucleus, it's converted to a circle. And circles are sometimes good things because they're pretty easy to replicate. The virus dissociates host nuclear domain 10s and re-enters whatever that space is that was filled with ND10s to form replication factories with both host and virus constituents. And it replicates as something that's called a rolling circle. So what does that look like? That looks like this. Here's your circular molecule. There's a nick. One strand gets pushed off. The other strand gets replicated. And the strand that gets pushed off, here's your 5' end, right? Here's your now gets replicated in this direction. So what happens is the circle rolls, and it rolls off these long lengths of genomic DNA. And these are also catenates. Those of you who know something about lambda DNA replication know that it replicates as catenated molecules. And these have to get cleaved at very specific places, and they get packaged. So that's a continuous molecular machine that is rolling out long loops of linear DNA. In order to do that, you need a variety of herpes simplex virus gene products. There are 3 that form a primase. So these are 3 different gene products that are required to form the primase activity. There's another that's a processivity protein, and that helps the DNA polymerase move through the viral DNA. We talked about UL9 at the beginning of the lecture, and that's its origin recognition protein, the one that sees the origin. UL29 is the DNA binding protein for herpes simplex virus. And finally, we have DNA polymerase. And you isolate all of these proteins, and you get the chemically pure, and you throw them into a test tube along with an appropriate origin of replication, and you get nothing. So it tells you that they're well, it doesn't tell you that they're necessary but not sufficient, but it tells you that they alone are not enough to replicate viral DNA. So what has been done is each one of these gene products has been obtained from, or has been cloned, the DNA that encodes it has been cloned. And those clones have been transformed into mammalian cells along with a plasmid that has an origin of replication. And DNA that's made in plasmids is cleaved by an enzyme called DPN1, which only recognizes methylated adenines. And mammalian cells don't have methylated adenines. They only methylate cytidine. And so if DNA replicates, then this plasmid molecule, this is a southern blot after cleavage with DPN, will no longer be cleaved. So this plasmid sequence will be copied, and you'll make thousands of copies of it, but only the original plasmid sequence from E. coli will be cleaved. So here it is thrown into a cell with any one of those 7 gene products left out. With any one of those 7 genes left out, you get no replication. If you have all of them in the mixture, that is a mixture of cloned fragments plus template, then you see replication. So this is termed DPN1 resistant, and that's how we know that these guys are important for replication. We just don't know all of the host components that are also important.